Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Endocrinology and Reproduction. In this particular lecture, we will go through another important endocrine gland, the pancreas, then change gears and move into the reproductive system and look how the endocrine organs play a role in the reproductive system. So let us begin. First, let us look into the pancreas and its hormones. The pancreas, in addition to its digestive functions, secretes two important hormones, insulin and glucagon, that are crucial for normal regulation of glucose, lipid, and protein metabolism. Next, let us look into the physiologic anatomy of the pancreas. The pancreas is composed of two major types of tissues, as shown in the schematic right here. The first are the acini, which secrete digestive juices into the duodenum, and the second are the islets of Langerhans, which secrete insulin and glucagon directly into the blood. The human pancreas has 1 to 2 million islets of Langerhans, each only about 0.3 millimeters in diameter, and organized around small capillaries into which it secretes its hormones. The islets contain three major types of cells: the alpha, beta, and delta cells, which are distinguished from one another by their morphological functions, by their morphology and staining characteristics. The beta cells, constituting about 60% of all the cells of the islets, lie mainly in the middle of each islet and secrete insulin and amylin, a hormone that is often secreted parallel with insulin, although its function is unclear. The alpha cells, about 25% of the total, secrete glucagon, and the delta cells, about 10% of the total, secrete somatostatin, which is the third important hormone that is secreted by the pancreas. Next, let us look into insulin and its metabolic effects. Insulin is a hormone that is mainly associated with energy abundance. Insulin secretion is associated with energy abundance. That is, when there is great abundance of energy giving foods in the diet, especially excess amounts of carbohydrates, insulin is secreted in great quantity. Thus, the stimulus for secretion of insulin is ingestion of very high energy containing foods such as carbohydrates. In turn, the insulin plays an important role in storing this excess energy that is taken in by the body via the diet. In case of excess carbohydrates, it causes them to be stored as glycogen, mainly in the liver and the muscles. Also, the excess carbohydrates that cannot be stored as glycogen are converted under the stimulus of insulin into fats and stored in the adipose tissue. In case of proteins, insulin has a direct effect in promoting amino acid uptake by cells and conversion of these amino acids into proteins. In addition, it inhibits the breakdown of the proteins that are already present in the cells. When insulin is secreted into the blood, it circulates almost entirely in an unbound form. It has a plasma half-life that averages only about 6 minutes, so that it is mainly cleared from circulation within 10 to 15 minutes. Next, let us look into the chemistry of the insulin molecule and its synthesis. Insulin is a small protein with a molecular weight of only 5808. It is composed of two amino acid chains connected to each other by disulfide linkages. Insulin is synthesized in the beta cells as an insulin pre-pro hormone. This initial pre-pro hormone has a molecular weight of about 11,500, but it is then cleaved in the endoplasmic reticulum to form proinsulin with a molecular weight of about 9,000. Most of this is further cleaved in the Golgi apparatus to form insulin and peptide fragments before being packaged in the secretory granules. Here we have a figure showing the structure of insulin, and here we can see the two disulfide bonds that are present in the insulin protein. In the pre-pro hormone state, the two ends there that we can see right here, ending with aspartate and threonine, are essentially connected by a loop structure, which is then cleaved off by by processing in the endoplasmic reticulum and the Golgi apparatus to finally form the finished in the final insulin protein, which is functional in nature. Next, let us look into activation of target cell receptors by insulin and the resulting cellular effects. To initiate its effects on target cells, insulin first binds with and activates a membrane receptor protein. As we have seen time and again throughout the endocrinology lectures, protein hormones which cannot pass directly through the cell membrane need to bind to a membrane receptor via which it signals to the cell. The insulin receptor, which is shown right here, is a combination of four subunits held together by disulfide linkages, which consist of two alpha subunits and two beta subunits. The insulin binds with the alpha subunits. The portion of the beta subunits protruding into the cell become autophosphorylated. This activates a local tyrosine kinase, which in turn causes phosphorylation of multiple other intracellular enzymes, including a group called insulin receptor substrates or IRS. In this way, insulin directs the intracellular metabolic machinery to produce the desired effects on carbohydrate, fat, and protein metabolism. So here we can see insulin binding to the alpha subunits of the insulin receptors, which in turn causes the beta subunits to be autophosphorylated. Next, the autophosphorylated beta subunits phosphorylate insulin receptor substances or IRS, which in turn begin a secondary messenger cascade, which in turn can play a role in glucose transport, protein synthesis, fat synthesis, glucose synthesis, and growth and gene expression. Next, we look into in depth in each of these effects of the insulin protein. First, we'll look into The effect of insulin on carbohydrate metabolism. Insulin promotes muscle glucose uptake and metabolism. During much of the day, muscle tissue depends not on glucose for its energy but on fatty acids. The principal reason for this is that the normal resting muscle membrane is only slightly permeable to glucose except when the fiber is stimulated by insulin. And thus, when insulin stimulates the muscle fiber, glucose can enter the muscles directly and thus can be used as a source of energy by the muscles. Next, insulin promotes liver uptake, storage, and use of glucose. One of the most important of all effects of insulin is to cause most of the glucose absorbed after a meal to be stored almost immediately in the liver in the form of glycogen. Then, between meals, when food is not available, the blood glucose concentration begins to fall. Insulin secretion decreases rapidly, and the liver glycogen is split back into glucose, which is released back into the blood. Insulin promotes conversion of excess glucose into fatty acids and inhibits gluconeogenesis or production of glucose in the liver. Next, lack of effect of insulin on glucose uptake and usage by the brain. The brain is quite different from most other tissues of the body in that insulin has little effect on uptake or use of glucose. Instead, the brain cells are permeable to glucose and can use glucose without the intermediation of insulin. This is because brain work functions mainly on glucose only. It does not use any other source of energy, and thus for its function, it does not depend on an intermediary like insulin for the signal to uptake and use glucose, but directly uses glucose from the blood. Here in this diagram, we can see intracellular glucose plotted against extracellular glucose when we have given insulin and against a control group. That is the amount of glucose present in the extracellular fluid versus intracellular fluid when there is glucose present and when there is not. So we see when extracellular glucose increases, in case of the control, the glucose stays mostly in the extracellular fluid, shown by the control line, which is almost parallel to the x-axis. But in case of insulin, the line has a sharp slope, which means most of the glucose is now being shuttled into the intracellular fluid, and thus cells of different types are uptaking glucose instead of letting it remain in the extracellular fluid. That is essentially the action of insulin. Next, let us look into effect of insulin on fat and protein metabolism. Insulin promotes fat synthesis and storage. First, insulin increases the utilization of glucose by most of the body's tissues, which automatically decreases the utilization of fat, thus functioning as a fat sparer. However, insulin also promotes fatty acid synthesis. This is especially true when more carbohydrates are ingested than can be used for immediate energy, thus providing the substrate for fat synthesis. Almost all this synthesis occurs in the liver cells, and the fatty acids are then transported from the liver by the way of blood lipoproteins to the adipose cells to be stored. Next, let us look into insulin, how insulin plays a role in protein synthesis and storage. During the first few hours after a meal, when excess quantities of nutrients are available in the circulating blood, not only carbohydrates and fats, but proteins are also stored in the tissues, and insulin is required for this to occur. The mechanism of this is as follows. Insulin stimulates transport of many of the amino acids into the cells. Insulin then increases the translation of messenger RNA, thus forming new proteins. Insulin also increases the rate of transcription of selected DNA genetic sequences. Insulin inhibits the catabolism of proteins or the breakdown of proteins. In the liver, insulin depresses the rate of gluconeogenesis or the production of glucose. Here in this diagram, we can see the plot of insulin secretion versus plasma glucose concentration. And we can see that as glucose concentration increases, insulin secretion also increases for a while before saturating at a very high value. This shows that insulin, the stimulus for insulin secretion is essentially glucose that is present in the plasma or the extracellular fluid. Next, let us look into control of insulin secretion. Increased blood glucose stimulates insulin secretion. At the normal fasting level of blood glucose of 80 to 90 milligrams per 100 ml, the rate of insulin secretion is minimal. If the blood glucose concentration is suddenly increased to a level 2 or 3 times normal and kept at this high level thereafter, insulin secretion increases markedly in 2 stages. Next, let us look into feedback relation between blood glucose concentration and insulin secretion rate. This response of insulin secretion to an elevated blood glucose concentration provides an extremely important feedback mechanism for regulating blood glucose concentration. That is, any rise in blood glucose increases insulin secretion, and the insulin in turn increases transport of glucose into liver, muscle, and other cells, thereby reducing the blood glucose concentration back toward the normal value. Next, let's look into glucagon and its various functions. Glucagon, a hormone secreted by the alpha cells of the islets of Langerhans when the blood glucose concentration falls, has several functions, of which the most important is to increase the blood glucose concentration, an effect that is exactly opposite to that of insulin. Like insulin, glucagon is a large polypeptide. It has a molecular weight of 3,485 and is composed of a chain of 29 amino acids. Now let us look into the effects or the functions of glucagon, first of which include effects on the glucose metabolism. Glucagon crosses glycogenolysis an increased blood glucose concentration. The most dramatic effect of glucagon is its ability to cause glycogenolysis in the liver, which in turn increases the blood glucose concentration within minutes. Glucagon also increases gluconeogenesis. Even after all the glycogen in the liver has been exhausted under the influence of glucagon, continued infusion of this hormone still causes continued hyperglycemia. This results from the effect of glucagon to increase the rate of amino acid uptake by the liver cells and then the conversion of many of the amino acids to glucose by gluconeogenesis. Next, let us look into regulation of glucagon secretion. The steps of regulation of glucagon secretions are as follows. Increased blood glucose inhibits glucagon secretion. Increased blood amino acids stimulate glucagon secretion, and exercise stimulates glucagon secretion. Next, let us look into somatostatin, which is the third hormone that is secreted by the delta cells of the pancreas, which inhibits glucagon and insulin secretion. The delta cells of the islets of Langerhans secrete the hormone somatostatin, a polypeptide containing only 14 amino acids, which has multiple functions as follows. Somatostatin acts locally within the islets of Langerhans themselves to depress the secretion of both insulin and glucagon. Somatostatin decreases the motility of stomach, duodenum, and gallbladder. Somatostatin decreases both secretion and absorption of the gastrointestinal tract. The principal role of somatostatin is to extend the period of time over which the food nutrients are assimilated into the blood. At the same time, the effect of somatostatin to depress insulin and glucagon secretion decreases the utilization of the absorbed nutrients by the tissues, thus preventing rapid exhaustion of the food and therefore making it available for a longer period of time. Next, let us look into an overview of the entire blood glucose regulation system. In a normal person, the blood glucose concentration is narrowly controlled, usually between 80 and 90 milligram per 100 milliliter of blood in the fasting person each morning before breakfast. This concentration increases to 120 to 140 milligrams per 100 milliliter during the first hour or so after the meal. But the feedback systems for the control of blood glucose return the glucose concentration rapidly back to the control level, usually within 2 hours after the last absorption of carbohydrates. Conversely, in starvation conditions, the gluconeogenesis function of the liver provides the glucose that is required to maintain the fasting blood glucose level. The mechanisms of achieving this is as follows. The liver functions as an important blood glucose buffer system. That is, when the blood glucose rises to a high concentration after a meal, the rate of insulin secretion also increases. As such, as as much as two-thirds of the glucose absorbed from the gut is almost immediately stored in the liver in the form of glycogen. Then, during the succeeding hours, when both blood glucose concentration and the rate of insulin secretion fall, the liver releases the blood glucose back into the blood. Both insulin and glucagon function as important control systems for maintaining a normal blood glucose concentration. When the glucose concentration rises too high, insulin is secreted. The insulin, in turn, causes the blood glucose concentration to decrease towards normal. Conversely, a decrease in blood glucose stimulates glucagon secretion. The glucagon then functions in the opposite direction to increase the glucose towards normal. Also, in case of severe hypoglycemia, a direct effect of low blood glucose on the hypothalamus stimulates the sympathetic nervous system. In turn, the epinephrine secreted by the adrenal glands causes still further release of glucose from the liver. And finally, over a period of 8 hours to days, both growth hormone and cortisol are secreted in response to prolonged hypoglycemia, and they both decrease the rate of glucose utilization by most cells of the body, helping to return the blood glucose towards normal. After looking into the summary of blood glucose regulation, let us look into the abnormalities or the disease conditions that can arise from abnormal control of blood glucose or secretion of pancreatic hormones. The most important of these include diabetes mellitus. Diabetes mellitus is a syndrome of impaired carbohydrate, fat, and protein metabolism caused by either lack of insulin secretion or decreased sensitivity of tissues to insulin. There are two general types of diabetes mellitus. Type 1 diabetes, also called insulin dependent diabetes mellitus or IDDM, is caused by lack of insulin secretion. Or type 2 diabetes, also called non-insulin dependent diabetes mellitus or NIDDM, which is caused by decreased sensitivity of target tissues to metabolic effects of insulin. This reduced sensitivity to insulin is often called insulin resistance. So first, let us look into type 1 diabetes, which is caused by lack of insulin production by beta cells of the pancreas. Injury to the beta cells of the pancreas or diseases that impair insulin production can lead to type 1 diabetes. Viral infections or autoimmune disorders may be involved in destruction of beta cells. The symptoms of type 1 diabetes are as follows. Blood glucose concentrations rise to very high levels in diabetes mellitus. Increased blood glucose causes loss of glucose in the urine. Increased blood glucose causes dehydration. Chronic high glucose concentration causes tissue injury. Diabetes mellitus causes increased utilization of fats and metabolic acidosis. Next, let us look into the type 2 diabetes, which is caused by resistance to metabolic effects of insulin. Obesity, insulin resistance, and metabolic syndrome usually precede development of type 2 diabetes. Type 2 diabetes, in contrast to type 1, is associated with increased plasma insulin concentration, which is known as hyperinsulinemia. This occurs as a compensatory response by the pancreatic beta cells for diminished sensitivity of target tissues to metabolic effects of insulin. Most of the insulin resistance appears to be caused by abnormalities of the signaling pathways that link receptor activation with multiple cellular effects. Impaired insulin signaling appears to be closely related to toxic effects of lipid accumulation in tissues such as skeletal muscle and liver secondary to excess weight gain. Now, after looking into the pancreas, we finally change gears and look at the reproductive and hormonal functions of male, following which we will look into the reproductive and hormonal functions of female. So let us get started with this part of the lecture. So first we're looking into the reproductive and hormonal functions of the male. First we look into the physiologic anatomy of the male reproductive organs. The first figure, or the figure A, shows the various portions of the male reproductive system. And the second figure gives a more detailed structure of the testis and the epididymis. The testis is composed of up to 900 coiled seminiferous tubules, each averaging more than one half meter long, in which the sperm are formed. The sperm then empty into the epididymis, another coiled tube about 6 meters long. The epididymis leads into a vas deferens, which enlarges into the ampulla of the vas deferens immediately before the vas enters the body of the prostate gland. Two seminal vesicles, one located on each side of the prostate, empty into the prostatic end of the ampulla, and the contents from both the ampulla and the seminal vesicles pass into an ejaculatory duct leading through the body of the prostate gland and then emptying into the internal urethra. Finally, the urethra is the last connecting link from the testis to the exterior. The urethra is supplied with mucus derived from large number of minute urethral glands located along its entire extent, and even more so from bilateral bulbourethral glands or copper glands located near the origin of the event. Next, let us look in detail in the process of sperm formation or spermatogenesis. During the formation of the embryo, the primordial germ cells migrate into the testes and become immature germ cells called spermatogonia, which lie in 2 or 3 layers of the inner surfaces of the seminiferous tubules. A cross section of one is shown in the figure 8, which is right here. So here is the seminiferous tubules and these are the interstitial cells. The spermatogonia begin to undergo mitotic division beginning at puberty and continually proliferate and differentiate to definite stages of development to form sperm as shown in figure B. So here we can see the spermatogonia that after going through the differentiation finally form the spermatids. Let us look into the steps of spermatogenesis. In the first stage of spermatogenesis, the spermatogonia migrate among Sertoli cells towards the central lumen of the seminiferous tubule. The Sertoli cells are very large with overflowing cytoplasmic envelopes that surround the developing spermatogonia all the way to the central lumen of the tubule. The next step is meiosis. Spermatogonia that cross the barrier into the Sertoli cell layer become progressively modified and enlarge to form large primary spermatocytes. Each of these, in turn, undergo meiotic division to form 2 secondary spermatocytes. After another few days, these 2 divide to form spermatids that are eventually modified to form spermatozoa or sperm. During the change from spermatocyte stage to the spermatid stage, the 46 chromosomes or 23 pairs of chromosomes of the spermatocyte are divided so that 23 chromosomes go to one spermatid and the other 23 to the second spermatid. This also divides the chromosomal genes so that only one half of the genetic characteristics of the eventual fetus are provided by the father, while the other half are derived from the oocyte provided by the mother. The entire period of spermatogenesis from the spermatogonia to spermatozoa takes about 74 days. Next, we look into the 6 chromosomes that are present in the sperms. In each spermatogonia, one of the 23 pairs of chromosomes carries the genetic information that determines the sex of each eventual offspring. This pair is composed of one X chromosome, which is called the female chromosome, and one Y chromosome, the male chromosome. During meiotic division, the Y chromosome goes to one spermatid that then becomes the male sperm, and the female X chromosome goes to another spermatid that becomes a female sperm. The sex of the eventual offspring is determined by which of these 2 types of sperm fertilizes the ovum. Here we have a schematic which shows us the whole process of spermatogenesis via a diagram. So here we can see how the process starts from primordial germ cells. It then converts to a spermatogonia. The spermatogonia then proliferates, undergoes several cell divisions to form the primary spermatocyte, which then undergoes the first meiotic diffusion to form the secondary spermatocyte, followed by the second meiotic division to form the spermatids, which finally differentiate to form the premature sperm. Next, we look into the formation of the sperm and its structures. When the spermatids are first formed, they still have the usual characteristics of epithelial cells, but soon they begin to differentiate and elongate into spermatozoa. As shown in the figure, each spermatozoa is composed of a head and a tail. The head comprises the condensed nucleus of the cell with only a thin cytoplasmic and cell membrane layer around its surface. On the outside of the anterior two-thirds of the head is a thick cap called the acrosome that is formed mainly from Golgi apparatus. This contains a number of enzymes similar to those found in lysosomes of the typical cell, including hyaluronidase, which can digest proteoglycan filaments of tissues, and powerful proteolytic enzymes, which can digest proteins. These enzymes play important roles in allowing the sperm to enter the ovum and fertilize it. The tail of the sperm, called the flagella, has three major components. First, a central skeleton constructed of 11 microtubules, collectively called the axoneme. The structure of this is similar to that of the cilia found on surfaces of other types of cells. And using the sperm moves using the ciliary movement that we have already discussed in the lecture 2 of the whole lecture series. A thin cell membrane covering the axoneme, and 3, a collection of mitochondria surrounding the axoneme in the proximal portion of the tail called the body of the tail. Back and forth movement of the tail or flagellar movement provides motility for the sperm. Next, let us look into hormonal factors that stimulate spermatogenesis. These include testosterone, secreted by the Leydig cells located in the interstitium of the testis, is essential for growth and division of the testicular germinal cells, which is the first stage of forming sperm. The luteinizing hormone, secreted by the anterior pituitary gland, stimulates the Leydig cells to secrete testosterone. Follicle stimulating hormone, also secreted by the anterior pituitary gland, stimulates the Sertoli cells. Without this stimulation and the conversion of spermatids to sperm, the process of spermatogenesis will not occur. Estrogens formed from testosterone by Sertoli cells, when they are stimulated by the follicle stimulating hormone or FSH, are probably also essential for spermiogenesis. Growth hormone, as well as most of the hormones of the body, is necessary for controlling background metabolic functions of the testes. Growth hormone specifically promotes early division of the spermatogonia themselves, and in its absence, as in pituitary dwarfs, spermatogenesis is severely deficient or thus causing infertility. Next, let us look closely at testosterone and other male sex hormones. First, let us look into secretion of testosterone by the interstitial cells of lady in the testis. The testis secrete several male sex hormones which are collectively called androgens, including testosterone, dihydrotestosterone, and androstenedione. Testosterone is formed by the interstitial cells of lady, which lie in the interstices between the seminiferous tubules. Next, let us look into the metabolism of testosterone. After secretion by the testis, about 97% of testosterone becomes either loosely bound with the plasma albumin or more tightly bound with the beta globulin or sex hormone binding globulin and circulates in the blood in these states for 30 minutes to several hours. By that time, the testosterone either is transferred to the tissue or is degraded into inactive products that are subsequently excreted. Much of testosterone that becomes fixed to tissues is converted within the tissue cells to testosterone, especially in certain target organs such as prostate gland in the adult and external genitalia of the male fetus. In the first schematic, we can see the interstitial cells of Leydig and the blood vessels and fibroblasts covering them, and the seminiferous tubules that are shown in orange. And here we can see the testosterone and dihydrotestosterone molecules that hormones that are present. Next, let us look into functions of testosterone. In general, testosterone is responsible for distinguishing characteristics of the masculine body. Even during fetal life, the testes are stimulated by chorionic gonadotropin from the placenta to produce moderate quantities of testosterone throughout the entire period of fetal development and for 10 or more weeks after birth. Thereafter, essentially no testosterone is produced during childhood until about ages of 10 to 13 years. Then testosterone production increases rapidly under the stimulus of anterior pituitary gonadotropic hormones at the onset of puberty and lasts throughout the most of the remainder of the life, dwindling rapidly beyond the age of 50 to 20 to 50% of the peak value by age of 80. Functions of testosterone during fetal development. Testosterone secreted first by the genital regions and later by the fetal testis is responsible for development of the male body characteristics, for example, the male sex hormones. The testes usually descend into the scrotum during the last 2 to 3 months of gestation. This is dependent on testosterone. Next is the effect of testosterone on development of adult primary and secondary sexual characteristics. After puberty, the increasing amount of testosterone secretion caused the penis, scrotum, and testis to enlarge about 8-fold before the age of 20 years. In addition, testosterone causes secondary sexual characteristics of the male to develop, beginning at puberty and ending at maturity. Next is the effect on the distribution of body hair. Testosterone causes growth of hair over the pubis, upward along the linea alba of the abdomen, sometimes to the umbilicus and above, on the face, usually on the chest, and less often on the other regions of the body, such as the back. It also causes hair on most of the portions of the body to become more prolific. Baldness, testosterone decreases the growth of hair on the top of the head. Effect on the voice. Testosterone secreted by the testes or injected into the body causes hypertrophy of the laryngeal mucosa and enlargement of the larynx. The effect causes at first a relatively discordant cracking voice, but this gradually changes into the typical adult masculine voice. Testosterone also increases the thickness of the skin and can contribute to development of acne. Testosterone increases the thickness of the skin over the entire body, increases the rate of secretion of body's sebaceous glands, which can lead to acne. Testosterone increases protein formation and muscle development. One of the most important male characteristics is development of increasing musculature after puberty, averaging about a 50% increase in muscle mass over that in female. Testosterone increases bone matrix and causes calcium retention. The bones grow considerably thicker and deposit considerable additional calcium salts. Thus, testosterone increases the quantity of bone matrix and causes calcium retention. Testosterone also increases basal metabolism. Injection of large quantities of testosterone can increase basal metabolic rate by as much as 15%. Effect on RBCs. When normal quantities of testosterone are injected to a castrated adult, the number of red blood cells per cubic millimeter of the blood increases 15 to 20%. Effect on electrolyte and water balance. Many steroid hormones can increase the reabsorption of sodium in the distal tubules of the kidneys. Testosterone also has such a function, but only to a minor degree in comparison to adrenal mineralocorticoids, whose main function is to increase the reabsorption of sodium in the distal tubules of the kidneys. Next, let us look into control of male sexual functions by hormones from the hypothalamus and the anterior pituitary gland. A major share of the control of the sexual functions in both male and female begins with the secretion of gonadotropin releasing hormone or GnRH by the hypothalamus. This hormone in turn stimulates the anterior pituitary gland to secrete gonadotropic hormones, which include the luteinizing hormone and the follicle stimulating hormone. In turn, LH is the primary stimulus for secretion of testosterone by testes, and FSH mainly stimulates spermatogenesis. GnRH and its effect on increasing the secretion of LH and FSH. GnRH is a 10 amino acid peptide secreted by neurons whose cell bodies are located in the arcuate nuclei of the hypothalamus. Then the GnRH is transported to the anterior pituitary gland in the hypophyseal portal blood and stimulates release of two gonadotropins, LH and FSH. The gonadotropic hormones LH and FSH both are secreted by the same cells called gonadotropes in the anterior pituitary gland. LH and FSH are glycoproteins. They exert their effects on the target tissues in the testes mainly by activating cyclic adenosine monophosphate second messenger system, which in turn activates specific enzyme systems in the respective target cells. Testosterone and its regulation of its production by LH. Testosterone is secreted by the interstitial cells of Leydig in the testis, but only when they are stimulated by luteinizing hormone. Next, we look into the inhibition of anterior pituitary secretion by LH and FSH by testosterone, which serves as a negative feedback control of testosterone secretion. The testosterone secreted by the testis in response to LH has a reciprocal effect of inhibiting anterior pituitary secretion of LH. Most of this inhibition probably results from a direct effect of testosterone on the hypothalamus to decrease the secretion of GnRH. This in turn causes a corresponding decrease in secretion of both LH and FSH by the anterior pituitary, and the decrease in LH reduces the secretion of testosterone by the testis. Regulation of spermatogenesis by FSH and testosterone. FSH binds to specific FSH receptors attached to the Sertoli cells in the seminiferous tubules. This causes these cells to grow and secrete various spermatogenic substances. Simultaneously, also has a strong tropic effect on spermatogenesis. Next, let us look into the negative feedback control on seminiferous tubule activity, which is the role of the hormone inhibit. When the seminiferous tubules fail to produce sperm, secretion of FSH by anterior pituitary gland increases markedly. Conversely, when spermatogenesis proceeds too rapidly, pituitary secretion of FSH diminishes. The cause of this negative feedback effect on the anterior pituitary is believed to be the secretion by the Sertoli cells of still another hormone called inhibit. This hormone has a strong direct effect on the anterior pituitary gland to inhibit the secretion of FSH and possibly a slight effect on the hypothalamus to inhibit secretion of GnRH. Inhibin's potent inhibitory feedback effect on the anterior pituitary gland provides an important negative feedback mechanism for control of spermatogenesis. Next, let us move on to the female hormones and female physiology before pregnancy. To start off this part, first let us look at physiologic anatomy of the female sexual organs. The principal organs of the human female reproductive tract, the most important of which are the ovaries, fallopian tubes, uterus, and the vagina. Reproduction begins with the development of ova in the ovaries. In the middle of each monthly sexual cycle, a single ovum is expelled from an ovarian follicle into the abdominal cavity near the open fimbriated ends of the two fallopian tubes. This ovum then passes through one of the fallopian tubes into the uterus. If it has been fertilized by a sperm, it implants into the uterus, where it develops into a fetus, a placenta, and fetal membranes, and eventually into a baby. Next, let us look into the female hormonal system. The female hormonal system, like that of the male, consists of three hierarchical hormones as follows: a hypothalamic releasing hormone, gonadotropin releasing hormone GnRH, the anterior pituitary sex hormone, or the follicle stimulating hormone FSH, and luteinizing hormone LH, both of which are secreted in response to release of GnRH in hypothalamus, the ovarian hormones estrogen and progesterone, which are secreted by the ovaries in response to the two female sex hormones from the anterior pituitary gland. Here we can see a schematic showing the female reproductive system. First, let us look into the monthly ovarian cycle and the function of the gonadotropic hormones. The normal reproductive years of the female are characterized by monthly rhythmical changes in the rates of secretion of female hormones and corresponding physical changes in the ovaries and other sexual organs. This rhythmical pattern is called female monthly sexual cycle, or less accurately, the menstrual cycle. The ovarian changes that occur during the sexual cycle depend completely on the gonadotropic hormones FSH and LH secreted by the anterior pituitary gland. Ovarian follicle growth, follicular phase of the ovarian cycle. The first stage of the follicular growth is moderate enlargement of the follicle itself. Then follows growth of additional layers of granulosa cells in some of the follicles. These follicles are known as primary follicles, which is about 12 of them. Development of antral and vesicular follicles. During the first few days of each monthly female sexual cycle, the increasing concentration of both FSH and LH causes the growth of primary follicles, manifested as proliferation of granulosa cells around the oocyte. The mass of the granulosa cells secretes a follicular fluid which accumulates till an antrum is formed. The early growth of primary follicle up to the antral stage is stimulated mainly by FSH alone. Then greatly accelerated growth occurs, leading to still larger follicles called vesicular follicles. This phase is regulated by FSH, estrogen, and LH. Once the antral follicles begin to grow, their growth occurs almost explosively. The ovum itself also enlarges in diameter another threefold to fourfold, giving a total ovum diameter increase up to tenfold. As the follicle enlarges, the ovum itself remains embedded in a mass of granulosa cells located at one pole of the follicle. Only one follicle fully matures each month, and the remainder undergo atresia. Here we can see a schematic which shows the entire process. Next, we look into ovulation. Ovulation in a woman who has a normal 28-day female sexual cycle occurs 14 days after the onset of menstruation. Shortly before ovulation, the protruding outer wall of the follicle swells rapidly, and the small area in the center of the follicular capsule, called the stigma, protrudes. In another 30 minutes or so, fluid begins to ooze from the follicle through the stigma, and about 2 minutes later, the stigma ruptures wildly, allowing a more viscous fluid which has occupied the central portion of the follicle to evaginate outward. This viscous fluid carries with it the ovum, surrounded by a mass of several thousand small granulosa cells called the corona radiata. And here we can see the ovum and the corona radiata around it. Surge of LH is necessary for ovulation. LH is necessary for final follicular growth and ovulation. Without this hormone, even when large quantities of FSH are available, the follicle will not progress to the stage of ovulation. Next, let us look at corpus luteum and the luteal phase of ovarian cycle. During the first few hours after the expulsion of the ovum from the follicle, the remaining granulosa and the thecus internal cells change rapidly into lutein cells. They enlarge in diameter 2 or more times, and become filled with lipid inclusions that give them a yellowish appearance. This process is called luteinization, and the total mass of cells together is called corpus luteum. The granulosa cells in the corpus luteum develop extensive intracellular smooth endoplasmic reticula that form large amounts of female sex hormones, progesterone and estrogen, which contain more progesterone than estrogen. In the normal female, the corpus luteum grows to about 1.5 centimeters in diameter by 7 to 8 days after ovulation. Then it becomes to involute and eventually loses its secretory functions in about 12 days after ovulation, becoming the corpus albicans. During the ensuing few months, this is replaced by connective tissue and over months is absorbed. Luteinizing function of the LH. The change of granulosa and theca internal cells into lutein cells is dependent mainly on LH secreted by the anterior pituitary gland. Secretion by corpus luteum is an additional function of LH. Next, let us look into the functions of the ovarian hormones, estradiol and progesterone. First, let us look at estrogens. In normal, non-pregnant females, estrogens are secreted in significant quantities only by the ovaries. Although minute quantities are also secreted by the adrenal cortices. Only 3 estrogens are present in significant quantities in the plasma of human female: beta estradiol, estrone, and estriol, of which the beta estradiol is the major one. Progestins. By far the most important progestins is progesterone. In the normal non-pregnant female, progesterone is secreted in significant amounts only during the later half of the ovarian cycle, when it is secreted by the corpus luteum. Synthesis of estrogens and progestins. They are synthesized in the ovaries mainly from cholesterol derived from the blood, but also to a slight extent from acetyl-CoA or acetyl coenzyme A, multiple molecules of which can combine to form the appropriate steroid nucleus. Here in this schematic, we can see the chemical structures of beta estradiol, estrone, and estriol, and also that of progesterone. Let us look into the functions of estrogen. A primary function of estrogen is to cause cellular proliferation and growth of tissues of the sex organs and the tissues related to reproduction. Effect of estrogen on uterus and external female sex organs. At puberty, the quantities of estrogen secreted in the female under influence of pituitary gonadotropic hormones increases 20-fold or more. At this time, the ovarian, fallopian tubes, uterus, and vagina all increase several times in size. During the first few years after puberty, the size of the uterus increases twofold to threefold. Estrogen causes marked proliferation in the endometrial stroma and greatly increased development of the endometrial glands. Similar changes occur in the fallopian tubes. The estrogens also initiate growth of the breasts and of milk producing apparatus. Effect of estrogens on skeleton. Estrogens inhibit osteoclastic activity of the bones and therefore stimulate bone growth. Effect of estrogens on protein deposition. Estrogen causes slight increase in total body protein. Effect of estrogens on body metabolism and fat deposition. Estrogens increase the whole body metabolic rate slightly, but only about 1/3 as much as the increase caused by male sex hormone testosterone. They also cause deposition of increased quantities of fat in subcutaneous tissues. Effect of estrogen on hair distribution. Estrogen do not greatly affect hair distribution. However, the hair does develop in the pubic region and the axillae after puberty. Androgens formed in increased quantities by the female adrenal glands after puberty are mainly responsible for this. Effect of estrogens on the skin. Estrogens cause the skin to develop a structure that is soft and usually smooth. Effect of estrogens on electrolyte balance. Estrogens like aldosterone and also some other adrenocortical hormones cause sodium and water retention in the kidney tubules. Next, let us look into the functions of progesterone and regulation of the female monthly rhythm. First, let us look into the effect of progesterone on the uterus. By far, the most important function of progesterone is to promote secretory changes in the uterine endometrium during the later half of the monthly female sexual cycle, thus preparing the uterus for implantation of the fertilized ovum. Effect of progesterone on fallopian tubes. Progesterone also promotes increased secretion by the mucosal lining of the fallopian tubes. These secretions are necessary for nutrition of the fertilized dividing ovum as it traverses the fallopian tube before implantation. Effect of progesterone on breasts. Progesterones promote development of lobules and alveoli of the breasts, causing alveolar cells to proliferate, enlarge, and become secretory in nature. Next, let us look into regulation of female monthly rhythm, interplay between ovarian and hypothalamic pituitary hormones. The hypothalamus secretes GnRH, which causes the anterior pituitary gland to secrete LH and FSH. Hypothalamic centers for release of GnRH. The neuronal activity that causes pulsatile release of GnRH or gonadotropic releasing hormone occurs primarily in the mediobasal hypothalamus, especially in the arcuate nuclei of this area. Next, let us look into the negative feedback effects of estrogen and progesterone in decreasing both LH and FSH secretion. Estrogen in small amounts has a strong effect to inhibit the production of both LH and FSH. Also, when progesterone is available, the inhibitory effect of estrogen is multiplied, even though progesterone by itself has little effect. These feedback effects seem to operate mainly on anterior pituitary gland directly, but also operate to a lesser extent on the hypothalamus to decrease secretion of GnRH. Hormone inhibin from corpus luteum inhibits FSH and LH secretion. In addition to feedback effects of estrogen and progesterone, other hormones seem to be involved, especially inhibin, which is secreted along with the steroid sex hormones by the granulosa cells of the ovarian corpus luteum in the same way that Sertoli cells secrete inhibin in the male testis. This hormone has the same effect in the female as in the male, inhibiting the secretion of FSH and to a lesser extent LH by the anterior pituitary gland. Positive feedback effect of estrogen before ovulation, the pre-ovulatory LH surge. For reasons not exactly understood, the anterior pituitary gland secretes greatly increased amount of LH for 1 to 2 days beginning 24 to 48 hours before ovulation. The cause of this abrupt surge in LH secretion is not known. However, several possible explanations are as follows. It has been suggested that estrogen at this point in the cycle has a peculiar positive feedback effect on stimulating pituitary secretion of LH and to a lesser extent FSH. The granulosa cells of the follicles begin to secrete small but increasing quantities of progesterone a day or so before the pre-ovulatory LH surge. And it has been suggested that this might be the cofactor that stimulates the excess LH secretion. Finally, let us look into the feedback oscillation of hypothalamic pituitary ovarian system. The feedback oscillation that controls the rhythm of the female sexual cycle seems to operate in approximately the following three events: post-ovulatory secretion of ovarian hormones and depression of pituitary gonadotropins. During this time, the corpus luteum secretes large quantities of both progesterone and estrogen, as well as the hormone inhibin. All these hormones together have a combined and negative feedback effect on the anterior pituitary gland and hypothalamus, causing the suppression of both FSH and LH secretion about 3 to 4 days before the onset of menstruation. Follicular growth phase. 2 to 3 days before menstruation, the secretion of estrogen, progesterone, and inhibin from the corpus luteum decreases to a low ebb. This releases the hypothalamus and anterior pituitary from the negative feedback of these hormones. At the time that the menstruation begins, pituitary secretion of FSH begins to increase again, several days after menstruation begins. LH secretion increases slightly as well. These hormones initiate new ovarian follicle growth and a progressive increase in secretion of estrogen, reaching a peak at about 12.5 to 13 days after the onset of the new female monthly sexual cycle. pre-ovulatory surge of LH and FSH causes ovulation. At about 11.5 to 12 days after the onset of the monthly cycle, the decline in secretion of FSH and LH stops abruptly. It is believed that the high level of estrogens at this time, or the beginning of progesterone secretion by the follicles, causes a positive feedback stimulatory effect on the anterior pituitary, which leads to a terrific surge in secretion of LH and to a lesser extent FSH. This pre-ovulatory LH and FSH surge leads to both ovulation and subsequent development of and secretion by the corpus luteum. This was all we had for today. These are the references for the slides, which are the chapter 78, 80, and 81 from the textbook of medical physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.